and to a large extent also did a lot of the work that was published with Peter Mombarts. Peter Mombarts, for those who know him, he liked to tell people what to do, but doing experiments, I think that's Fan's credit to do all the analysis of the mice, but also she herself made some really interesting contribution to the search, the genetic search for the role of olfactory receptors in establishing the map of olfactory neuron projection to the olfactory bulb. So she did the beautiful PhD work with Richard, and then switched to a different system as a postdoc in Mark Tessier-Lavine's lab, where first she became interested in the somatosensory system, which is still her main interest now, but from a developmental standpoint. And in Mark's lab, who at the time was at Stanford, I believe, she identified some really interesting patterning mechanism of somatosensory neurons. So based on this work, she got a job at Duke University as an assistant professor. And here her research first started by continuing the work on the developmental aspect of the somatosensory system, but then quickly switching into more coding issue or circuit organization of the somatosensory system, both how the connectivity is established between neurons in the somatosensory system, what is the type of connectivity being established. She also branched out and looked at more motor aspect of whisker movement, in particular with David Kleinfeld, and then made some really extremely interesting contribution on the aspect of pain related to the somatosensory system, and some of this she will tell us today. So in addition to this really very creative work on the somatosensory system, she also showed an interest in developing tools, genetic tools in particular, and some of those, like the Kane system, have been widely adopted to look at neurons that are activated in particular tasks. And when I went to Duke last time, remember basically everyone around you was using the Kane system. So that ended up being just a really interesting new tool that Fan developed. So overall, very creative, very rigorous research, and then all these fabulous genetic tools. And today, she will tell us about some recent work that she has done that is absolutely fascinating. The title of her talk is General Anesthesia, Sleep, and Pain Suppression. Thank you, Fan.
You can hear me now? Okay. Thank you, Catherine, for the really sweet introduction. I came to US and I only speak a little bit English, and Catherine taught me some French. I still the only 3 words I remember is salut, merci, and Mambatilapa. I have no idea that of other things, but it was very useful when I go to Paris last year. I used the first 2 words aloud. I always admired Catherine. I've been following her models and trying to be creative and then do interesting things, find my own niche, not to stepping on other people's toes. And so that has been what I learned from Catherine. So I'm going to tell you briefly about Kane, if I can find my mouse. Yeah, I think some of you know it. I just gonna, for those of you who do not know the technology, I'll give a brief introduction. Then I'm going to talk about how we use this to study general anesthesia, particularly 2 aspects of general anesthesia. One is the relationship of general anesthesia with sleep. The other is with analgesia. So Kane is a technology we developed. Basically, it's a Faust-based method. Faust is the immediate early gene in mammals. We use a lot to stain for activated neurons. And at the time when we made the tool, there are Faust TTA and false Cree-ER. However, in our hands, a lot of regions, we just simply cannot use these tools to label the activate neuron. So we decided to make our own tool. So this really is by putting the destabilized TVA receptor after the last amino acid of false gene with a 2A peptide in between. So the idea is then when the cells express a lot of false, it will also transiently express this receptor. Then we can use a pseudotyped virus, in this case a lentivirus coated with a pseudotyped code to infect these neurons and bring in any gene we wanted. So far we're most successful using this cane to bring Cree into activated neurons. So at the time, in order to test this tool, we have to infect the neurons express, let's say, something red color, and then do the behavior again to induce false gene expression again, so that if the red cells are colocalized with green false positive cells by the same behavior again, then we would know the method works. So we were not a lab studying innate behavior, but at the time we decided to choose the innate behavior to do this test, mainly because we figure innate behavior, you would more likely to have the same neurons that activated by the same behavior. So this is the resident intruder test, and I think we use it in Catherine's lab also. The male interaction, the aggressive male, when another male comes in, we would say get out, right? Try to beaten up the other male, and then so this male will show defensive and escape behaviors. And after 15 or so such interactions, you stay in the brain to look for where false is expressed, and obviously it's in many, many places. And we're only going to focus on this hypothalamic region, ventral medial hypothalamus, and ventrolateral division. You can see two stripes of cells in this region that are highly false positive after in the intruder mice who is being beaten up. So we call these social fear neurons. So we use a cane to capture these cells. Then we let the mouse being beaten up again to induce false again. And you can see a really nice colocalization. It's not 100%. It's always about 60 to 80%. But I think that's as good as it gets compared to other methods. So we can capture these cells. And then to do functional manipulations, we would express channel rhodopsin in these so-called social fear cells to test whether or not we can then drive these fearful behaviors, so staying away from the other mouse. So these mice will then be used as a resident. In other words, it shouldn't be afraid of any intruders, but we are going to use channel rhodopsin to turn on these hypothalamic cells that we captured using cane, and then see what is his response to the intruder. Okay. Now, when we published this work, we were only using unilateral stimulation, but subsequently we did a bilateral stimulation, which is the videos I'm going to show you, and it's way more robust. So this is a male-male interaction. You can see he's beating up the other one because it's his own cage. Then the light comes on, he stays at the corner, stop being aggressive, and even show some defensive pose. And then as the lights comes off, he will think about it. This is my own cage. I smell my own shit. I should be aggressive. So he go back to beating up the other one, right? So this is a pretty robust. And then you turn the lights on, immediately hide to the corner. So this is a quite robust behavior. And when the lights off, he'll go back to beat up the other male again. So we can toggle these behaviors very efficiently with using this optogenetic tools. Now, we even can activate these cells in response to females. So usually male mouse chase a female doing this typical mouse pornography. And then we will then turn the lights on to activate these social fear cells. You can see immediately escape from the female. And, whoa, you're a scary woman. And then he will hide in a corner and then think about it. And then the lights off, go back to chase the female again, right? So it says these cells indeed can drive robust conspecific fear. So this is the tool we developed, and we also made a rabies version so we can trace the presynaptic input. Suffice to say, for the purpose of this talk, you just need to know CAIN is a method that we can use to label and manipulate strongly false positive neurons. It works moderately when it's a moderately expressed false. Okay, I have to say that it's a limitation. If it's weak false, then we can't capture them. Okay, general anesthesia. So this really is mostly because of my interest in pain, and I wanted to figure out a way to suppress pain without losing consciousness. So I went into anesthesia knowing very well that there are two obvious sort of characters of general anesthesia. One is the sedative effect, right? Put you to sleep. The other is make you feel no pain. This way, surgery can proceed. So the first part would be related to this, the sedative effect of general anesthetics. Now, in EEGs, you can easily distinguish an active brain, awake brain, with a high frequency oscillation but very small amplitude versus a sedated or sleeping or unconscious brain with these high amplitude oscillation, which are called third oscillation or slow wave oscillations. Interestingly, there are many chemically extremely diverse general anesthetics. I'm listing a few of these from studies of Patrick Purdon and Emery Brown's work. And you can see propofol, sevoflurane, these are GABA drugs. Ketamine is an NMDA antagonist, and dexamethasone is alpha-2 adrenergic receptor agonist. They cause different brain oscillations. And so, like propofol has alpha oscillation, and then ketamine has gamma oscillation. So this has led to the idea different anesthetics may create this loss of consciousness using different mechanisms. On the other hand, they also share a feature, which is a strong amplitude of slow wave oscillation at 0.5 to 4 hertz. Okay, and slow wave oscillation is also a signature of the non-rapid eye movement sleep or deep sleep. So this shared feature makes us wonder whether or not there is then a shared neural substrate underlying these strong third oscillations in anesthetic state and in sleep. We further hypothesize if there is a shared substrate, this shared substrate is activated by diverse anesthetics. So we're looking for an active mechanism rather than a passive mechanism. So to do this, we did a simple experiment, put mouse under isoflurane for 2 hours, and just stained for false to see where in the brain there's still remaining activate neurons. So here they are. The first region that we caught our eye is this super strong cluster of cells. So this is optic chiasm. It's at the corner of the optic chiasm, and this is the supraoptic nucleus. Okay, now so this is a control brain. You can see scattered false expression. And after 2 hours, the brain is really quiet, very limited false expression. So these remaining false pulses stands out in the brain. Now, supraoptic nucleus is very famous, a region famous for, they are really the hub of neuroendocrine cells that relieve hormones into the blood. So indeed, when we did in situ hybridization, so this is false activated by isoflurane, and this is vasopressin, dynorphin, oxytocin, and galanin. So what you can see is about 80% of false positive cells are vasopressin positive. They're not the oxytocin cells. The vasopressin cells, most of them also express dynorphin. So they're essentially dynorphin and vasopressin co-releasing neuropeptidergic neurons that are known to release things into the posterior pituitary then throughout the body. So about 80% are vasopressin dynorphin cells. So initially, I thought this must be an artifact. There must be other things turn on false rather than activity, right? I mean, serum starvation can turn on false. So I made my students do just a wired recording into the SON region, see if there's any evidence that in vivo there are activated neurons. So he did a random recording. Indeed, there are units. So you can see these units. Okay, these are just even raw trace. This is isoflurane exposure. This unit has baseline activity and gets suppressed. Whereas this unit was pretty quiet, then increased firing upon isoflurane exposure. So indeed, there are these suppressed unit and then isoflurane activated unit. In fact, the activated unit activated before the state transition into the unconscious state. So this is shown by strong delta oscillation and no muscle movement. So this is the unconscious state. So these cells fire before transition and stop firing before the mouse wake up or after isoflurane was stopped putting into the system. So this is just a summary. Most of cells are suppressed. Not surprisingly, the brain under anesthesia, are becoming quiet, but there are subsets of cells that are strongly activated. So then it's a population. Not all of them are vasopressin. We decide to use cane to capture all of them. So we put the mouse under anesthesia, use a cane to capture the cells, and then later on re-expose them to isoflurane, and you can see a really nice, efficient and specific capturing of these cells. Most of them are in this region, in the supraoptic nucleus, but there is also a scattered, which we call para-SON region. So once we can efficiently capture these anesthesia-activated cells, we can do the fun experiment to re-expose them to different anesthesia. Remember, all the cane captured cells are captured based on isoflurane. Okay, then we re-give them mouse isoflurane, propofol, ketamine, or dexamethasone. So all different drugs activate neurons in SON and vasopressin cells, and they're very good co-localization or overlap. So this really is a region that represents a commonly activated neural substrate by all these diverse anesthetics. In fact, we patched onto these cells in slice and then give them isoflurane, propofol, ketamine, and dexamethasone. You can see all drugs depolarize these cells instead of hyperpolarizing them as in controls, which I'm now showing here. We are just starting to do in vivo phototagged recording. So we put channel rhodopsin in these cells and put a tetra drive here, try to hunt these cells by light. So if the cells are responding to light, like this unit, you will see if the video plays. So we only have 3 units like this. So each time we'll have light pulses, you can see the spikes. So it's a photo identified unit. So it is one of the isoflurane activated cells that we captured with cane and express channel rhodopsin. And we can then basically record the same cell in response to isoflurane. Here's the EMG that's showing the state transition. And what you can see is so this isoflurane comes on, the cell start to fire, and the blue spikes are the units, and you can see the cell fire more and more. The mouse still moving, and eventually the mouse will stop move, and that's the loss of conscious state, and then the cell will continue to fire. So indeed, these cells in vivo fire by exposure to isoflurane and before state transition. So that led us to say, okay, maybe they are indeed doing something. So we then used a chemogenetic tools to activate these cells. Initially, we did the chemogenetics because these cells spread about 1.2 millimeters at the base of the skull. So we thought optogenetic maybe just not cover enough space. So we express the excitatory dreads into these cells and give them CNO and see what happens. So this is a control M-cherry mouse that with a CNO injection, which you can see mouse sleep really short bouts. They don't sleep very well. So when there's a strong delta oscillation, very little muscle movement, this is when they're in sleep and or slow wave sleep. And the effect of the drug of activating these neuroendocrine cells is really to extend the sleep duration. Okay, so this is quantified here. They double the amount of sleep, but this is mostly from increasing the per bout duration, not bout number. So in other words, these cells are not switched. So mouse still switch into sleep the same amount of number, but they just sleep longer when they get into sleep. In fact, we also did experiment with the vasopressin Cre, which is problematic Cre because the homozygous lethal. But still, even with this not perfect tool, we can see just turning on the vasopressin cells, you extend the duration of sleep by extending the bout number. So why do you want a neuroendocrine system to extend sleep, right? Under what normal circumstances do we sleep longer? So that naturally brings the idea of sleep drive or sleep deprivation. When you didn't sleep yesterday, today you sleep longer, something like that. So we did a sleep deprivation experiment and then let the mouse immediately sacrifice after sleep deprivation or let the mouse recover the sleep. It turns out the neurons are activated by sleep pressure, but they're actually gradually turned off when the mouse actually caught up their sleep. So it's in the sleep pressure pathway. So the idea is then you have an endocrine system, release peptide, and that then signal longer, and that coordinate the system to sleep better. So we then did the optogenetic stimulation. So we briefly give about 3 minutes of optogenetic stimulation, and then afterwards we turn off the lights and see what happened to the mouse. So because it's in the optic chiasm, light alone in GFP mice induce robust wakefulness, and mouse actually run around. They're uber awake. But despite the artifact of light-induced wakefulness, once we turn off the lights in the channel rhodopsin mice, they can sleep for 10 to 15 minutes, and really with a strong delta power. So that is says the lasting effect of peptidergic signaling continue to drive sleep. Okay. We did a loss of function experiment. We killed these cells with diphtheria toxin, DTR and then diphtheria toxin. So what happens is really, so this is normal sleep. You have a lot strong delta wave, and the delta wave just crashes. Okay, so we can score these as sleep, but they're light sleep. They don't sleep very well. And in fact, they're so sleep deprived. A lot of the mice died in this paradigm. So you do need these cells for endogenous sleep to give you a strong drive to sleep better. We finally did anesthesia experiment. If we briefly activate these cells, then expose isoflurane. So what happens is duration under anesthesia is longer. So they take much longer to emerge. However, the induction is unaffected. So the switch is unaffected. It's just this duration. And if we silence these neurons with archaeorhodopsin, the mouse emerges from anesthesia much quicker. So this is interesting. So these neurons not only release peptide into the pituitary to your blood, but also have lots of dense core vesicles, and then their dendrite cover the hypothalamus. So they have massive dendritic release. So I actually think it's their release into the hypothalamus as well as into the cerebrospinal fluid that then signal long distance, coordinate the whole brain into this global sort of sedated state, and then keep the brain in that state longer. So this paper is just published online in Neuron, so yeah, you can check it out and give me feedbacks. So now I'm gonna turn to analgesia, right? So most of you probably think, of course you don't feel pain if you don't have any consciousness left. You don't perceive pain. However, there are these interesting reports of intraoperative awareness. So most of the intraoperative awareness are awareness of surgeons' talk, conversations, even knowing things are operating. You can feel the sensation, but you dissociate that from pain. So that says you could have a sensation that is dissociated from ascribing that to pain, and you still have consciousness under anesthesia. So in fact, ketamine, low-dose ketamine, has been used to treat pain. If no other drugs works, they usually give you ketamine. You still have sensation. You still can sense something intense, but you don't ascribe that as pain. So there is a dissociation mechanism somewhere in the central nervous system that you can block you from thinking a sensation is pain. So we're really interested in finding the central pain or central analgesic circuit, pain suppression circuit. And again, we thought this must be an active mechanism. We were looking for where in the brain, other than SON that I just talked to you, that also general anesthesia activate neurons. And we find them in central amygdala. So here shows the central amygdala. This is control. And in fact, you can see this. I'm showing you isoflurane and ketamine. And it's true for propofol and dexamethasone. Okay? So there are strongly anesthesia-induced false positive neurons. And these are all GABAergic neurons showing by a GABA marker. Now, central amygdala has been known for its role in fear as well as in processing pain. In fact, we had a paper on central amygdala, parabrachial to central amygdala pathway for affective pain. So it's kind of bizarre to see anesthesia activate cells in the central amygdala. We did a lot of markers. People study CEA, study somatostatin, dynorphin, neurotensin, and these are not colocalized with anesthesia-activated false positive cells. There are partial overlap with PINK1, which is a preproenkephalin. It's an endogenous opioid, and partial overlap with PKC-theta. So suffice to say, we haven't found any marker that absolutely perfectly colocalized with anesthesia-activated cell. Therefore, we turn to our one-trick pony cane to capture these cells and see what happens to these cells. So this actually works really efficiently. This is Faust, this is cane, and you can see beautiful colocalization. So we managed to label these cells very effectively. And we expressed GCAMP in these cells and did a miniscope imaging with green lens. So the mouse is moving around. Isoflurane will be infused. You will see a red line here. That's when isoflurane comes in. It's a 1%, so it takes a little bit of time. And you can see this is processed. This is raw video. As the animal eventually will stop moving, go down under anesthesia, right? And then the cells will continue to fire. So their sets of cells continue to fire. It is a heterogeneous activity. You can see lots of activities throughout anesthesia. And if we sort these cells based on the second half versus first half of activity during anesthesia, you can see a small subsets are persistently activated under anesthesia, but the larger subsets are transiently activated by general anesthesia. And there's a small number are suppressed, which we think are just background labeling. The technique is not perfect. You always label some background false expressing cells. But suffice to say, there are cells that are activated by isoflurane. And the same isoflurane activated cell, if we image under ketamine, you can also see ketamine sustained activate cells and ketamine transiently activate cells. Okay, so now that we confirm they are anesthesia, either transiently or sustainably activated cells, we wonder what their function in normal behavior. Initially, we thought, could it be fear? So we activate these cells using channel rhodopsin by using cane to expressing channel rhodopsin in these cells. And so the lights is already on, you probably can't see it. But the mouse really move around. There really isn't any freezing or cornering behavior. If anything, the mouse explores the inner parameter a bit more and does a lot of rearing, which is a behavior of exploration. And so these are quantified here. Compared to GFP, no difference. We also did the elevated plasmase to test anxiety. And again, there's no anxiety-like behavior when we activate the anesthesia-activated cells in central amygdala. So then I told my student to give mouse pain and see what happens. So we can either inject formalin, which is super nasty, to the cheek or to the paw. And the mouse will rub the cheek or lick the paw. It's their typical, when you have a sustained pain, they will have these recuperative behavior. Then I'm going to show you what happens without light and with light. So this is a mouse. Oops. Oh no, it's thinking. It's my thing probably crashed. No, it's not. Okay, good. Okay, let's see again. Please play. No, it's not playing. It was playing before. Sorry. And it wouldn't let me escape. My computer shows a sign. Crap. Come on. Sorry, I don't know what's going on. It was, let me just escape this and show. I wouldn't let me quit the show.
If you wait a few seconds and then get out of the presenter view, then maybe you can show the video that way.
Yeah, so it would not let me get out of presenter view. So it crashed right now. Like I couldn't, it shows a little circle on my thing.
So force quit and...
Force quit, okay. All right, so it did force quit. Okay, let's restart this again. Okay, so let me go to... You really need to see the video to believe that I'm not. Okay, so let's see if it plays here. Okay, so I'm just gonna play not in presenter view. Okay, so it's rubbing, and the light will come on in middle 10 seconds time, and you can see stop rubbing immediately, right? So it will immediately stop rubbing the inflamed cheek. And I'm going to show you this. Hopefully it works. This is licking the foot. And then when the light comes on to activate the CEA cells, see, it just work around, no problem. So stop these behaviors immediately. So really it's quite robust in terms of behavior. And I had 2 different people repeating this, and it was all very true. And that's quantified here. So we then did a silence experiment. See what happens if we silence these cells. Do they feel more pain? Okay, so just a quick reminder, when you inject formalin, each time you give channel activating cell, they stop licking. But gradually the mouse itself will stop licking because it just needs to guard the inflamed paw. And they can deal with it. At this time, so he stopped licking, guarding the paw, and moving around. Okay, then we're going to turn the lights on to using ER to inhibit the CEA cells, immediately resume licking, as if the pain comes back. Okay, so that's quantified here. So far I showed you channel rhodopsin activating the CEA and GA, we call CEA, GA cells, suppress pain, and your inhibiting cells exacerbate the pain. So what about the noxious induced, just simple reflexes? So we did a heat, cold, and mechanical in naive mice, normal mice. So every time we channel rhodopsin activating these cells, their sensory threshold went up, meaning take much stronger heat for them to withdraw, whereas the ER inhibition will take them much shorter time to withdraw. So this is for heat, and this is for dry ice. And in fact, for dry ice, you can see normally they quickly respond. They will move away. This is already channel rhodopsin. So usually it should be 2 seconds, but when we put it on, you can see he's even rubbing his face. Forget about it. His paw is doing that. So that's quantified here. I didn't show you the other control movie. So really, when you activate these cells, can suppress their thermal sensation. And this is the electronic Wangfrey. You poke them with this machine. Again, same bidirectional regulation. And this is a phase Wangfrey, poking at the phase. Now, GFP mice with or without light have the same withdrawal frequency. Channel rhodopsin mice will withdraw much less when we turn the lights on, whereas archaeorhodopsin mice will even respond to something that they normally don't respond. So I'm going to show you this. So this is a GFP control, and 0.4 gram usually do not causes any response. So this is below or their sensory threshold, okay? And no response, right? You can see. Now, so this is E-arch stimulation off. You probably can see the fiber poking. Now we're gonna turn the, later on we're gonna turn the lights on. My movie is not, so this is really, this is stimulation on with a 0.4 gram that they didn't respond before. Now you can see they respond, right? When this is a prototypical response with this head shake, and that's a slow motion. And as you repeatedly moving it, they really hate it. They will use a paw to get rid of that. So essentially, we can turn into an innocuous sense into a painful sense by silencing these neurons. And we also showed silencing these, if you pair a chamber with silencing these neurons, they will hate it. They will move to the other chamber, and they will have a lasting aversive memory. So that's summarized here. So this then made us wonder, there must be a basal level activity. And I actually showed you some basal level activity when I imaged for any seizure. So these basal level activity must be there to set the general threshold so you don't overreact to your environment. You can feel normal. You don't feel everything's pain. So what happens when we give them painful stimuli? So we image these neurons with, again, cold, heat, or mechanical stimulation. So I'm showing you an episode, four trials of poking at their face and imaging these cells. And mouse, when you poke them, they kind of sweated and run. So it's actually quite diverse. So these are the different Wangfrey fibers. And if you look at the activity, it's spread across the entire trial. So this is a withdrawal time where you can align them to stimuli onset. There really isn't any obvious population level activity. It's pretty flat, okay? So they're not time locked to stimulus with a few exceptions. So there are 6 cells we find have the stimulus related sort of turn on response. Majority of them are unchanged, and a few of them have sort of a suppressed activity, and that is quantified here. And this trend is generally true. Majority of them are unresponsive to dry ice or heat or Paul Wangfrey, with a few cells turn on and a few cells turn off. So in other words, the ongoing activity are set by some sort of internal state, and these cells are not responsive to pain activation, which is corroborated by Kane capturing these cells and then give them formally and to induce fouls, and you can see they're largely spatially segregated. So again, these imaging studies suggest that these cells are really your internal expectation, internal sense of how your body should interact and respond to physical world. So you're now super sensitive. So finally, we wanted to know whether or not when we activate these cells, we can also affect chronic pain. So what we do is we ligate this nerve, it's called infraorbital nerve, to the whisker pads. And this is the model for spontaneous and then hypersensitivity to the face after you ligate nerve. It's a classic neuropathic pain model, and they would have spontaneous wiping. And when we activate the CEA cells, the spontaneous wiping drastically reduces. We can stop them from doing that. And this even has a long-lasting effect. Even during post-stimulation period, they still have a reduced wiping behavior. We then tested with Wang Frey. So the mouse becomes hypersensitive. So they start to even respond to poking at a much lower gram range. And I'm going to show you the response to 1 gram, okay? 1 gram is super nasty under neuropathic conditions, like needle poking to them. And what you see is, nope. Sorry, my videos are not doing very well today. Okay, so this is GFP control. You poke at 1 gram, they hate it, okay? Sweat it off because it's just super nasty. And they will do this 100% of time. And you can turn the lights on to GFP. They will still swap it off because it's our control. It's supposed to do that. Okay, and so this is a nasty stimulant. Now, this is channel rhodopsin in CEA. Stimulation off, absolutely hated. Okay, 1 gram. Always try to get rid of the stimulation. Now we're going to turn the lights on. Same animal, lights on. And you can poke all you want, and it would not respond. Okay, so the effect is so drastic and so stunning. I had 2 people repeat this experiment, and I got convinced, and there was like 8 animals all show the same effect, right? So you can completely block not only hypersensitivity, but sensitivity. See, they are less sensitive. And come to think about it, it makes sense because you ligate the nerve. They shouldn't respond to peripheral stimulation, right? So why are they sensitive? I mean, if you look at the control side, they still respond to 1 gram because that's spinal reflex, okay? So that reflex kicks in. So the lesion nerve cannot respond to peripheral stimuli. Therefore, all the hypersensitivity must be somewhere in the brain. And if you can suppress the brain abnormal processing, then you can suppress the chronic, it's all in the brain, the chronic pain. And we did a place preference. In chronic conditions, the channel rhodopsin animal do prefer the stimulated side, presumably due to pain relief. These cells project widely throughout the brain. So these are just showing you a few examples in frontal cortex, nuclear accumbens, ventral BNST, basolateral amygdala, the association cortex, periaqueduct gray, peribrachial, even all the way to the brainstem and spinal cord. Although most projections are ipsilateral, if you look at the contralateral side, I'm showing you on the amygdala, there are also fibers in the contralateral side. So they are mostly ipsilateral projection, but the subset goes to the contralateral, and that's why we're seeing the bilateral effect. We did do rabies tracing, and they receive input from a wide variety of regions, insular, some entorhinal input, so we're still trying to figure out which of these regions and how do they set this ongoing activity, and again, whether or not it's involved in sort of pain expectations down the road, so other ongoing experiment. So in summary, general anesthesia activate an ensemble of neurons in the central amygdala. Activation or inhibition of these neurons can bidirectionally modulate both reflexive and affective behaviors. And activation of these cells really suppress sensitivity and hypersensitivity in the neuropathic pain model. So we're really dying to see if we can find other molecules other than anesthetics can activate these cells. That will be our next generation painkiller, but we haven't figured out how to do that yet. So this is my lab. We always do hot pot party, and this is my funding source. And I thank you very much for listening.
I have a question that I thought maybe you were going to get to in that very final slide. Are there any afferent inputs controlling from brainstem area that often sets alertness and
Yeah.
sleep control, and because if I understand correctly, you see this as maybe a final common pathway control center regardless of body need or for anesthetics. It all gets to here. So-
Yeah, so it does get input. So, for example, peribrachial, this is the alert center.
Ah,
Now,
okay.
so peribrachial goes to the pain activated cells as well as goes to anesthesia activated cells. So it's actually really interesting. Whereas if you look at the pain, only fast turn on in the pain activated cells, right? But not in these cells. I have a theory why that is. I actually think low stress are aggravating pain, whereas death-threatening stress, you want to run away. It only has to have super high level of stress, then you activate an analgesia cell. So this is so-called soldiers on battlefield.
Yeah.
When you lose an arm, you don't feel pain. You continue to fight for your life, right? So I think these input does send threatening signal, but their input must be weak. Only when it's super strong, then you activate analgesia activated cells. And they also receive input from periventricular thalamic region, which is, again, it's a region for a lot of salient features. So we are actively investigating these regions to see how that sets the cells.
That's the input, but the output,
Yeah.
what do you think these cells are controlling?
Yeah, so in the cingulate and prefrontal cortex, right, and nuclear accumbens,
But those are not the most major.
not the most major, right? So I really think the most major output, one region is this basolateral amygdala. That is really, so this is overexposed because at low MAC that's the labeling region. And so that's why at high MAC you can see on the other side. So I think BLA is one of the major output. And also this region, look at this little here. This is the insular, posterior insular temporal association region. I think insular plays a strong role in pain intensity. And then the BNST maybe for some valence. And also it goes back to peribrachial. So it does project back, probably to suppress pain. So these are, and then this region in the reticular region, okay, in the spinal cord, in the brainstem. So this region hosts a hub of cell that does something called DNIC, diffuse noxious inhibition. So when one arm is in pain, the rest of the body feel less pain because you want to focus on this arm. So this region suppresses pain in the rest of the region. So the cells project to that region also. So I think these regions are really interesting candidates to further work on. Yeah. Now.
So all the anesthetics that you tested in STRICE experiment,
Yeah.
experiment activated these neurons,
Yeah,
but
in SON.
these
Yeah.
anesthetics have very different actions.
Yes.
What do you think is the mechanism?
Yeah. So far, I only have synaptic blocker for isoflurane. So I block all synaptic transmission, still depolarize. For others are just direct patching. I initially tried to use GTACR, the chloride channel, light active to silence these cells. And these cells are activated. So in both the SON and in central amygdala, the cells have higher internal chloride concentration. They're really easy to reverse. Chloride becomes depolarizing rather than hyperpolarizing. Most of the anesthetic drugs are GABA agonists. So I think chloride definitely play an important role. I just don't have the porphyry patch working yet to really tease that out. But I do think at that level. And then if you have some other mechanisms in the circuit level that you somehow have local higher GABA, these cells get depolarized. It's actually really interesting. Yeah. Yes.
I'm curious, do you find sexual dimorphisms in the activation pattern in response to isoflurane anesthesia or something like that? Because there are sex differences in pain, chronic pain.
Yeah. So in our assays, so far we don't see any sexual dimorphism. We haven't delved into the certain models that are known to have sexual dimorphism. But the trigeminal pain is pretty uniform across sex. Maybe the TMJ pain has a sexual dimorphism. Just haven't worked on that.
I have a question related to the first population you described
Yeah.
AVPs.
Yeah. They're the source, right? Yeah, responsive. Yeah.
What are they connected to?
Yeah.
Also, I mean, you assume they're just playing with neural loop involved, but these cells, the PDN and SON, they are also sending projections.
Yes.
And also, what input do they receive? How are they associated with the sleep circuit in general? Do you have a sense?
I don't have a very good sense other than whatever stress I give animal, these cells turn on. So not just, people know that if osmolarity increase, right, you activate vasopressin, so you preserve water. And during sleep, in humans, you also have increased vasopressin expression so that you don't go to bathroom during the middle of night, something like that. But in anesthesia, I've talked to anesthesiologists, vasopressin is massively released. They know that, but they always ascribe that as stress. Because you're going to a surgery, you're stressed. So these cells are very easily activated by a lot of stress. But they didn't think anesthetics.
Stress keeps you awake.
Yeah, the stress keeps you awake, right? So that's why these are the sleep drive. So after stress, you really need to rest, sleep longer to recover, to have the benefit of. So that's why I think these cells then turn off actually during sleep. So I do think they are there to tell the body that, okay, when you finish what you have to do, you really need to recover sleep. So that's what I think what they're doing. Yeah. Yes.
So does this in some of neurons have to do with that endorphin production in the context of pneumonia?
Yeah, so my cells are in the CEA have a partial overlap with enkephalin, which is endogenous opioid. We did try an naloxone experiment in our behavior assay, see if naloxone, which blocks opioid action, can block the CEA anti-pain function. So at 5 mg per kg, it's already very high dose. We don't see the effect. But when we do 20 mg per kg, we can block it. But 20 mg, you block opioid receptor everywhere, right? So you block. So it may not be the evidence that these cells that are primarily using endogenous opioid. But I think they do release enkephalin, but they're mostly GABA. So I think they have the fast actions through GABA transmission. Yeah.
Since this is a lentiviral infection in this neuronal population, and you say that 80% of them are infected.
Yeah, with a CRE and CRE-dependent AAV.
Yeah. So
Yeah.
have you done a quantitation, like what is the minimum number of neurons required for this function? And if so, why is there a redundancy in the circuit?
So I haven't done a quantitative because we usually, the cane is pretty efficient. In anesthesia, it's almost like the perfect method for it because the brain is super quiet, almost no background. You just capture the cells. There they are. I confess I did try TREP2, leaching loss, false CRE-ERT2. I thought it'd be much easier. I don't have to do stereotaxic injection. I got 40 to 50% of cells with the TREP2 method. So I'm gonna try that, see 40 to 50 is enough to see an effect. So TREP2 in our hands works, but not as efficient as cane. Yeah, so usually 40%, but maybe 40% is enough to have effect. Yeah.
Okay. Any more questions? Thank you.
Thank you. Sorry for the video fiasco. Don't know what happened.
It ended up working with your sous chef.
I know, yeah.